Hello everyone, welcome back to Study Room. In this video, we will be studying about the asexual mode of reproduction, and we'll be knowing the different types of reproduction that actually come under the asexual mode of reproduction. Now let us begin with an example. As you can see here, these are the two photos that are given here. You can see this is a dough that is kept here for a long time, and if you keep it it keeps growing gradually. You can see there's this growth of size with time. So why this growth of size takes place? Because there's this presence of yeast that is present here, and it grows or reproduces and multiplies with the with gradually with time, and hence there's this growth of dough that takes place. Okay, also there's this picture of bread. You can see here there's these pieces of bread, and these are the kind of growth that takes place on bread, and these are the bread mold. These are the kind of fungi that grows on bread, and which is known as bread mold, and these fungi grow gradually with time when the bread grows stale. Okay, so when the bread is stale, you can see this growth of bread mold on the pieces of bread if you keep them for a long time in open. Okay, now how does these growths take place? In this video you can see that there's this fission taking place where a cell is getting divided into two different cells. Also you can see the budding of hydra that is taking place, that there's this small bud formation on the body of hydra, and that bud finally grows into a new hydra. Also you can see the mode where there's this regeneration in starfish. When if a body part of the starfish is cut off, or there's this injury in the body of starfish, it can easily regenerate that part in its body. Also you can see there's this mode of reproduction in plants, where a part of the plant, if it is grown in soil, then it grows into a new plant, and this is known as vegetative propagation. So talking about the different kinds of asexual reproduction, we can actually form a flowchart with it. Okay, and we can discuss these all forms of reproduction. As we saw fission taking place where a cell was divided into two different daughter cells. The fragmentation where the different fragments of an organism grow into different organisms, like different new organisms. Okay. Next is budding, where you have seen the budding that taking place in hydra, where a small bud growing into the body of hydra grows into a new organism. Next is vegetative propagation, where a part of a plant or a part of an organism grows into a new individual, and that is especially taking place in plants. Also we can see this regeneration that takes place generally in lizards. When the tail of a lizard is cut off, it grows into like the lizard can again grow that tail. Okay, and also we have seen the example of starfish, where the starfish can regrow its parts. We have the last one, that is spore formation, where the new individuals are formed from spores. Also, what do we mean by asexual reproduction? All the modes of reproduction that allow new generations to be created from a single individual is called asexual reproduction. So while defining asexual reproduction, you definitely need to mention that there is only single parent. Okay, so this is one of the most important points that you need to mention in the definition of asexual reproduction. Now, one by one, we will be talking about the different types of asexual reproduction, and the first type of asexual reproduction is fission. So, many bacteria and protozoa simply split into two equal halves during cell division. Okay, and the parent cell divides into daughter cells. It is further divided into two parts, that is binary fission and multiple fission. So you can see here that if one cell divides into many different cells, or it splits into two equal halves, then it is called as fission. And this fission happens in many bacteria and protozoa. Talking about the two types of fission that takes place, the first one we'll be talking about is binary fission. If a mother cell divides into two daughter cells, and each daughter cell begins its life as new individual, then this kind of reproduction is known as binary fission. Okay, and for example, it takes place in amoeba. Okay, so you have to define binary fission where a single parent amoeba, you can see here that a single parent amoeba is getting divided into two daughter cells, and there's this elongation of nucleus that takes place, and then the division of cytoplasm eventually takes place, and there's this formation of two daughter cells, and this happens in some of the organisms, and the most suitable example for this kind of binary fission is known as amoeba. Okay, so binary fission happens in amoeba, and the definition of binary fission is when a parent mother cell divides into two daughter cells, and each daughter cell begins its life as new individual. This is known as binary fission. In some unicellular organisms, there are different kind of organization in their bodies, like in leishmania. Okay, there's this one example called leishmania. Okay, so in leishmania, what happens is they have a whip-like structure at one end of the cell. Okay, there's this. If you take this is a leishmania. Okay, and there's this whip-like structure in one end of the cell. And in these kind of cells, what happens is the binary fission occurs in a definite orientation in relation to these structures. Okay, so the binary fission may happen this way. There can be a transverse fission, or there can be a longitudinal fission. So these kind of organisms have different kind of binary fissions, which depends on the orientation of their body, and hence you can also give example of leishmania in binary fission. Okay. Next, moving on to multiple fission. Okay, so what is multiple fission? If a single celled organism divide into many daughter cells simultaneously, this kind of fission is known as multiple fission. And the example of multiple fission can be the malarial parasite, that is known as Plasmodium. So Plasmodium is the parasite that causes malaria, and in this the fission that occurs, the mode of reproduction is multiple fission. You can see here there is this parent cell and there is this nucleus, and there is a cyst formation where many nuclei are contained in the cyst, and this cyst breaks and daughter cells are released. So there is this, from one cell many daughter cells are getting are being reproduced, and hence this kind of a fission is known as multiple fission. Okay, talking about fragmentation. Fragmentation includes breaking of parent organism into two or many fragments. Each fragment develops into an individual organism. For example, Spirogyra simply breaks up into smaller pieces upon maturation, which grow into new individuals. Okay. Now you can see here in this diagram, you can see here this is the diagram of Spirogyra, and the Spirogyra gets fragmented into two different parts, which is here you can see unequal fragmentation takes place. Like a part is small and a part is large, and then this both the parts can regenerate. Okay, both the parts can actually form the new individual, the new Spirogyra, and hence this kind of reproduction that takes place is known as fragmentation. Okay. Next up, we have regeneration. So regeneration is about the ability to give rise to new individual organisms from their body parts. Okay, so if the organism is somehow cut or broken into pieces, each part grows into complete individuals. So regeneration is carried out by specialized cells. These cells proliferate and make large number of cells. Okay, so due to the presence of these specialized cells or regenerable cells, so what these cells do, they actually help to grow the body parts which are lost. Okay, and thus an individual has the ability to give rise to give rise to the injured body parts. Okay, for example, we have simple animals like Hydra and Planaria, which can be cut into any number of pieces, and each piece grows into a complete organism. You can see here how it happens. If you cut a planaria into two halves, then both the parts of planaria can actually regenerate and they can form new organisms. Okay. Next up, we have budding. Organisms such as Hydra use regenerative cells for reproduction in the process of budding. A bud develops as an outgrowth due to repeated cell division at one specific site. These buds develop into tiny individuals and when fully mature, detach from the parent body and become new independent individuals. You can see here that how a Hydra, this is the Hydra, okay? This is a small formation of, this is a formation of bud. The bud has specialized cells which can regenerate, right, and form a new individual. So you can see that the bud gradually grows, keeps on growing, and then it detaches from the main body of the Hydra and forms a new Hydra. So this is how, with the help of an example or with the help of this diagram, you can see that how budding takes place in Hydra. The next is spore formation. So the thread-like structures that develop on the bread or the hyphae of the bread mold known as rhizopus. Okay, the bread mold that we saw in the first slide, in that you could see that there was growth, there was certain kind of growth, isn't it? There was certain kind of growth in a piece of bread, and with which you can say that the bread was stale. So these growth are caused by fungi known as bread mold, and the scientific name is called as rhizopus. Okay. The blobs are sporangia, which contain cells or spores that develop into rhizopus individuals. Okay, so the main body have spores or have sporangia, which contain cells or spores that develop into new rhizopus organisms. They are not reproductive parts. On the other hand, the tiny blob on a stick structure are involved in reproduction. The blobs are sporangia, which contain cells or spores that can eventually develop into new rhizopus individuals. The spores are covered by thick walls that protect them until they come into contact with another moist structure and can begin to grow. So you can see here how the spore formation occurs. There is this is the kind of the fungi that we have, and if you magnify it, you will find these kind of structures. Okay, these kind of structures, and in this you can find the structure called as sporangium, which will have so many spores, and these spores are the mode of reproduction. Okay, you can see here these are the spores. From sporangium the spores are formed, and these spores actually give rise to new individuals. Next up, we have the vegetative propagation. As you can see, there are two types of vegetative propagation. The first is natural vegetative propagation and second is artificial vegetative propagation. Natural vegetative propagation is usually happening with the help of plant parts just such as roots, stems and leaves. But in artificial vegetative propagation, there are different methods that are used like cutting, layering, grafting, or tissue culture. So these are the artificial methods that are used for vegetative propagation that usually takes place in the growth of or the reproduction of plants. Okay, vegetative propagation is the ability of plants to reproduce by bringing forth new plants from existing vegetative structures. So you can see from existing vegetative structures from the different parts of the plant, when they give rise to new plants, such kind of a reproduction is known as vegetative propagation. As you can see here, the reproduction is taking place from leaves or roots or stem. So these kind of reproduction is called as vegetative propagation. Next up, we have natural vegetative propagation, where you can see how the roots, by roots we can produce new plants. It happens in sweet potatoes. So for the example, we can mention sweet potato as the example for natural vegetative propagation by roots. Okay, you can see here this is a potato, and these roots grow and then they form new kind of individuals, new plants. So this is how vegetative, natural vegetative propagation takes place with the help of fruits in sweet potato. Next up we have leaves. So you can see how the leaves, how the leaves are instrumental in producing new organisms, new plants from them. And this is an example of bryophyllum, where the buds produced in the notches along the leaf margin of bryophyllum fall on the soil and develop into new plants. So these buds which are developed on the notches of the bryophyllum leaf, these buds will, when they fall on soil, they develop into new plants. Also, we can say this is the most common practice that we do for reproducing plants, for growing new plants. That is, we cut the stem of the plant, we take a branch and we place it in the soil, and this is how a new plant grows from that stem. So this is how we can actually see that these are the three kinds of vegetative propagation that are termed as naturally, that can be done naturally, that is with the help of roots, leaves and stem. Now we'll be seeing the pros and cons of vegetative propagation. Like, what are the advantages and disadvantages of vegetative propagation? You can see here the pros. Okay, so what are the advantages? The advantages are vegetative propagation is an easier method than growing plants from seeds, and vegetative propagation is used in plants which do not produce seeds. So you can see here that it is a very easy method, and it can be used in plants which do not produce seeds. So the plants which do not produce seeds, in those plants, how those plants reproduce? They reproduce through vegetative propagation. And what is the disadvantage or what is the cons of vegetative propagation? Parent plants with diseases transfer their diseases to the offspring. Okay, so the plants produced by asexual reproduction are short-lived than the plants produced by sexual reproduction. So when the plants are produced by seeds or by means of sexual reproduction, then those plants live longer. But these plants which are grown with the help of asexual reproduction, they are short-lived. Okay. And secondly, if the parent plant has any kind of disease, okay, then that disease is transferred to the offspring plant as well. So these are the two disadvantages or the two cons that we have for vegetative propagation. Now we will be talking about the artificial vegetative propagation. In artificial vegetative propagation, it is a method that is used such as layering or grafting to grow many plants like sugarcane, roses, etc. for agricultural or commercial purposes. Okay, so these are the methods, these are the specified methods that are used for growing plants like sugarcane, roses, etc. for agricultural purposes. And you can see here how these kind of things happen. These are the processes, these are the methods. The first is cutting. If you cut a plant and you place it, and then this plant, you cut the branch of a plant, okay, and then you place it in the soil or in a tub, and then this plant grows, okay, the root grows here. Second one is budding. You can see here how a part of the plant is taken, and then it is placed in, then it is placed, and then this part of the plant again grows. Okay. Next is grafting. You can see here how how a part of a different plant is cut, and then it is placed in another plant. Okay, and then these two, both the plants grow together and they form a hybrid kind of a plant. So this is the kind of these are the methods that are used for artificial vegetative propagation, which are generally used for agricultural or commercial purposes, right? Next we have tissue culture. So new plants are grown by removing tissue or separating cells from the growing tip of a plant. Why we are taking a growing tip of a plant? Because in the growing tip we have meristematic tissues. As you might have studied, that meristematic tissues are the tissues which help in the growth of plants. So these tissues are present in the growing tip. That is why we take cells from the growing tip of the plant. The cells are then placed in an artificial medium where they divide rapidly to form a small group of cells, and this small group of cells is called as callus. Okay, so you need to remember this term, which is very specifically used in tissue culture, which is known as callus. And callus is a small group of cells. The callus is transferred to another medium containing hormones for growth, okay, and differentiation. The plantlets are then placed in the soil so that they can grow into mature plants, okay. So this callus is then transferred into a medium where hormones for growth and differentiation are present. So you might have heard about the hormones that are important for the growth and differentiation of plants. We had studied those hormones before. So those hormones are present in that medium, and in that medium we place the callus, and then the callus grows into small plantlets, okay. They grow into small plantlets, and then these plantlets are again placed in the soil so that they can grow into mature plants. Using tissue culture, many plants can be grown from one parent in disease-free conditions. So this is a very important thing, that using tissue culture we can produce disease-free plants, and this technique is commonly used for ornamental plants, okay, the plants which are used for decoration. So in those plants this kind of technique is used. Let's understand this method of tissue culture with the help of this diagram. So in the first place, we will be taking a tissue sample from the parent plant. Okay, we will be scrapping a sample from the parent plant, and then we will be putting that tissue sample into a agar growth medium containing nutrients and auxins. So auxins are hormones that are helpful in plant growth and development, and the medium that we are taking is agar medium. Okay, so agar is a very important medium which has nutrients, and this helps in growth of the tissue that is placed in this medium. Okay, and then the samples develop into these samples when they are kept in agar medium. They get nutrient and auxins, and they grow into tiny plantlets. Okay, so they are growing into tiny plantlets, and these plantlets are then planted into compost. Then these plantlets are transferred into the soil and to grow new individual plants. Okay, so these new individual plants will grow from here when they are transferred into the soil. So that is how this whole process of tissue culture takes place, and it has several advantages, like it produces disease-free plants, and it is really a very good method for growing ornamental plants. So that is how tissue culture takes place. Now let's discuss the InText questions one by one. The first question is, how does binary fission differ from multiple fission? Binary fission differs from multiple fission in three different ways. That is, in binary fission, an organism divides into two similar organisms. But in multiple fission, an organism divides into two or more organisms. Okay, so when there are two or more organisms, then we have multiple fission. In binary fission, a cyst or thick layer is not formed around the cell, but in multiple fission, a cyst or a thick layer is formed around the parent cell, right? And the binary fission takes place in favorable conditions, but the multiple fission takes place in unfavorable conditions. Also, we have to mention the examples that we have for binary fission and multiple fission respectively, that is amoeba and paramecium for binary fission, and malarial parasite known as plasmodium for multiple fission. So this is how we'll be we'll be drawing a tabulated format for this for these three differences, and then mentioning the example that we have. Okay, next up our question is, how will an organism be benefited if it reproduces through spores? An organism is benefited by reproducing through spores because spores are surrounded by a thick layer which protects them in adverse conditions. So when the favorable conditions occur, these spores start to grow again. In this way, they are successfully alive in unfavorable conditions. So when unfavorable conditions happen, okay, unfavorable condition happens, what happens is There's this spores and they have a thick layer. Okay, these spores have a thick layer around them. But when the favorable condition happens, okay, so these spores, the thick layer breaks and these spores are further released into the environment and they can grow into new individuals. So that is how this is the benefit that the thick layer, surrounding the spores helps in protecting it in adverse conditions. Okay. Now we have the question number 3. Can you think of reasons why more complex organisms cannot give rise to new individuals through regeneration? In complex multicellular organisms, specialized cells make up tissues, tissues make up organs, organs make up organ systems, and finally organ system make up organisms. Since complex multicellular organisms have a very high degree of organization in their body, they cannot be reproduced from their cut body parts by the process of regeneration. For example, if we talk about a dog is a complex multicellular organism which cannot be regenerated from its cut body part. Say a cut tail, okay. So this is because the cells present in the cut tail of a dog cannot produce dog's organs like heart, brain, lungs, stomach, intestines and limbs, etc. needed for the making of a complete dog. So since we have specialized tissues for making different organs, that is why that is why we, the multicellular organisms, cannot actually grow new individuals through regeneration. Next question we have is, why is vegetative propagation practiced for growing some types of plants? Vegetative propagation is practiced for growing such plants which usually do not produce seeds or produce non-viable seeds. Okay, so the plants which do not produce seeds are the plant which produce seeds which are not used for growing new plants. So in those plants we can use these technique of vegetative propagation and hence those plants can be reproduced. The next question we have is, why is DNA copying an essential part of the process of reproduction? DNA copying, as we all know, is essential part of the process of reproduction so that the characteristics of the parent organisms are transmitted to its offspring, and at the same time some occasional variations are also produced in the offspring. The changes in the copy of DNA provide an organism the capability to survive in changing conditions. So hence DNA copying is a very essential part of the process of reproduction. That's all for this video of asexual mode of reproduction. Thank you.